Natural killer cells, NK cells, have the pivotal ability to detect and eliminate infected and malignant cells. Besides, due to their natural intrinsic killing capacity, and in the recent years, the development of genetically modified NK cells has enabled a new approach for cancer immunotherapy. Different strategies have been developed to engineer NK cells with a chimeric antigen receptor, and these CAR NK cells have an improved recognition of specific targets on cancer cells and gain an antigen-specific anti-tumor cytotoxicity. To date, however, the majority of CAR trials uses CAR T cells, but CAR NK cell trials are on the rise with 19 ongoing studies worldwide. In 7 of these trials, the NK cell line NK92 is being tested, and almost all of these trials are being conducted at different sites in China, with one exception. There's one phase 1 study under lead and supervision of Winfried Wells and Joachim Steinbach at our University Hospital in Frankfurt that address CAR-NK for treatment of HER2-positive glioblastoma. Then primary NK cells are evaluated in the other 12 ongoing clinical CAR-NK cell trials. There are mostly cytokine-expanded peripheral blood or cord blood-derived NK cells are tested, and few of these studies are being conducted in the US, one in Singapore, and the majority again in China. What I want to highlight is that Katie Redzvani and colleagues have just published data from the first 11 patients suffering from relapsed or refractory CD19-positive B-cell malignancies that were treated during first phase 1 clinical study with CD19 CAR-NK cells. 8 of these 11 patients showed a clinical response that occurred within 30 days after infusion. And I am impressed by the fact that CAR-NK cells persisted and were still detectable for at least 12 months in the patients monitored. Almost no cytokine release syndrome or neurotoxicity or GvHD has been reported for CAR-NK cells so far. One really important next step that needs to be envisioned is the planning of a multi-center phase 2 study to broadly confirm the clinical efficacy of these CAR-NK cell products.